Hi, I'm Mel Graham from the University of Minnesota, and I'm delighted to be presenting today thanks to the invitation from NJABR and the North American 3Rs Collaborative, some of our work coming out of the Preclinical Research Center. So today I'm going to talk through some of the objectives that we hope to with the laboratory animal sciences programs to talk a little bit about the role of how we design our experiments and the way we select our models and how that influences animal welfare and how it actually impacts our scientific outcomes in a major way and how those two are intertwined. I'm going to talk through some example strategies using behavioral management to intentionally introduce variation for generalizability of these translational programs or reduce some of the confounding to make our experiments more robust. So my research really is focusing on metabolic disease and immunology and transplantation, and particularly diabetes. And I always like to describe why this is important, right? Because when we talk about using animals, and particularly when we talk about using primates, the focus of this talk, we really want to understand what the relation to benefit is. So when we think about diabetes, in particular type 1 diabetes, what we recognize is this is a serious disease. Insulin isn't a cure for diabetes. It just keeps our patients alive until we find one. This disease is remarkably prevalent. 422 million adults have diabetes, and that is rising. And along with that comes a lot of complications that we see for these patients, ranging from stroke to cardiovascular disease to kidney disease. And this is highly expensive to the U.S. healthcare system. We have 30 million Americans that are being treated annually at a cost of $327 billion per year. So when we think about what kind of models that we're targeting to really move some of the work that we're looking at in discovery basis into our clinical therapeutics that benefits our patients, we start looking at the beginning. We look at our in vitro models or in silico models to screen. And as we walk towards our large animal models towards translation, we anticipate that translational relevance increases. But that spectrum is blurring, as we see, and you'll see certainly from other speakers in this conference, that really what drives the model is less about sort of the spectrum that we're all so used to, but really looking at different aspects of validity that drive the selection of these models. The first being face validity. So does the model itself have similar physiology? In the case of diabetes, we often think about things like hyperglycemia or glucose intolerance. Is the construct validity accurate? So is there similar underlying biology or etiology of disease? So for example, in type 1 diabetes, we're looking at beta cell loss. And this is something to think about as you're looking at the model, because when we talk about if we're looking at beta cell loss from the prevention perspective, the chemical surgical induction of diabetes violates that autoimmune construct that we typically see in our small animal models. And sort of the holy grail when we think about selecting models is really predictive validity. So does the model have a similar response to clinically effective interventions or therapeutic agents? And I'm going to use immunosuppression as a good example of this. And I wanted to also highlight some sort of ways in which we think about how these different areas, so for example, face and construct validity, can really tackle some of the problems that we see. And one of the things that's relevant, at least in diabetes, is we know that differences exist in insulin sensitivity and fat depots between females and males. So you'll see that there's metabolic control differences in males versus females under equivalent treatment. So again, we have to look to what's our experimental outcome that we're interested in and look at that again, similar physiology or underlying biology as we select that model. Also, as we think about what our animal models can do for us as we think towards translation, is we can look at what's happening on the clinical side, where we see things like underrepresentation of males in clinical obesity evaluations, despite knowing that obesity is prevalent equivalently between sexes. So this is a way that we can balance in our animal cohorts in the design phase to really unravel some of those sex-driven treatment outcome effects. So why do we really look at primates? Why is that a focus of one of the lectures here today? And I think, again, what we talked about is at least conventionally, in vitro and rodent studies support pathway determination or our mechanistic studies. That's really where we lean heavily on those two modalities, where NHPs, we get into examining practicality, safety, and generalized efficacy. And what we know is that there's this high relevance to the clinic. And why is that? Because of our close genetic relationship. And you see, of course, on that image to the side, that, again, you're sort of looking at how divergent you are along the evolutionary spectrum. And because of that close genetic relationship, we have remarkable anatomical, behavioral, developmental, physiological, and reproductive similarities with primates. And also, MHC expression, with a few exceptions, is very similar. We see a high level of conservation in amino acid sequences and immune system complexity. And that image that you see there on the side is demonstrating that with respect to something very current to us right now with COVID-19 susceptibility. That, again, is that conservation of amino acid sequences. Those bullet points that you see there on the left are what lead to what you see on the right. So we have high cross-reactivity with many human-directed biologicals in primates. There's, of course, some exceptions to that. Again, like I just mentioned, susceptibility to viral infections is high between primates and humans. In the case of COVID, again, we're seeing ACE2 gene and translated protein sequences that are strongly conserved across these primates. And again, you see that more so in old world macaques versus, for example, marmosets. And one of the things that I find most exciting, and I wanted to really focus in this talk, is when we're working in our primate models, is we can have a clinical trial like preclinical safety and efficacy study. So we can actually parallel some of the really elaborate therapies that we're thinking about for patients and the outcome measures that we're going to look at in patients in our primates. So you have this essentially pre-phase 0 that you're doing in monkeys. So how does this look, at least in what I was referencing earlier, when we think about immunology studies or transplantation studies? Primates are a very small fraction of the animals that we use in this research, right? So 1/3 of 1%, and that's really important to keep in mind. But again, they're an incredibly important step when we think about studies that have meaningfully informed how we walk into the human and how we walk into the clinic. And at least for immunosuppressive drug development, primates have been almost exclusively a gatekeeper in walking into clinical transplantation for the last 40 years. So one of the things that is really kind of interesting when you think about that is not only the forward translation that's been successful, but as we start to think about how we deal with things when they don't go the way they think. So you see this nice figure of walking through the human, through the in vitro, through the rodent, into the monkey, and then backwards again. So when we think about this idea of reverse translation, we can really get into this idea of how are our methods working? How can we do better? And I think clinical trials are a remarkable way for us to really take our cue from and think about how we do better at this early phase. So clinical trials focus a lot on uniform criteria that inform inclusion and exclusion, shared SOPs across trial sites, statistical analysis plans, and agreements on clinically relevant endpoints. Another piece of a good clinical trials design are really thinking about strategies that reduce design-imposed confounding. So again, barriers to adherence for patients and patient burden. And finally, reduced variability to achieve higher power with smaller numbers is always a consideration, and certainly should be a consideration when we're thinking about non-human primates. So when we look at that clinical experience and we think about it as a target to reverse translate back into our primate studies to improve them, there are a couple of factors that we know really affect rigor in the clinical situation. And this is obviously very well studied. And the first being non-adherence. So when we see that there's non-adherence in the clinical patient, we see increased variance, lowering of study power, and a reduction in the magnitude of our treatment effect. The second major factor is inappropriate subjects to start, so confounding our safety and efficacy interpretation. And as we think about these factors and we break them out into sort of treatment, patient, and factors related to the disease itself, on the treatment side, pain and distress associated with that. So treatment imposed restraints on the patient, limitations on sociality that the study might impose, or a big one is the frequency of medical intervention. So the frequency of that treatment leading to dropout or things like that. On the patient factor side, the desire for control and preference, it certainly plays a role into adherence. So control over one's environment, control over one's, again, disease course to some extent and how they manage it. And then, of course, there's the factors that just relate to the disease itself. So we know that our patients with chronic disease report some of the lowest quality of life measures that actually result from the disease state. And one thing to keep in mind for those of you that are using animal models of disease, these very highly representative animal models of disease have significant welfare implications that we need to keep at the forefront of our mind. So one of the things that I like to keep in mind is that when we think about our model design, the more intentional we are about it, it's our first and best defense. It's study rigor and animal welfare. So again, improving our likelihood of benefit, improving likelihood of successful translation. And there are many things that go into the state of our animals that I wanted to summarize here. We talked about how sickness and disease, so the actual model itself certainly factors in. A big one for those of us that are, again, at that safety and efficacy side of use in primates, the medical surgical interventions themselves impose substantial burden, but also just being a research subject, of course. So the handling and the environment that we impose on our research subjects and the way that it affects their sociality, all of those factor into their behavioral, psychological, and physiological health. And one of the things that I think is really key to keep in mind also in using primates is their heterogeneity is really a strength of the model, right? So when we think about translational strengths, we don't want to drive that piece of it out. And certainly the ability to perform interventions the way that we anticipate with our clinical patients is also a strength. And again, bringing that back to the forefront really allows us to refine our methods and think about how we can reduce that burden. So as you think about that piece of talking about what influenced success or failure in the clinical trial situation, we can connect that using behavioral management. There's certainly the disease state factor. So for example, in the situation that I described, when we have diabetic monkeys, for example, you are going to see that impose inherent harms. But we can certainly address the aspects of what we consider avoidable suffering, and that is allowing some determination or preference and thinking about motivation, so addressing more than just what the animal needs, but also their wants, to give an opportunity for them to exercise choice and control, particularly over their environment, and to use a pre-randomization or roll-in period to have our animals learn study procedures in low-stress, positive reinforcement conditions. And this allows us to also look at our animals' ability to comply and cope with the complicated treatment regimen. So as we talked about, making sure that that enrollment makes sense, that we're not enrolling what would normally be considered an ineligible animal. And then the piece that is also critical as you think also of the caregivers that care for our animals day to day is this relationship building. So thinking about the frequency and quality of the interactions, so that influences, of course, the satisfaction and the adherence of our animals with the complexity of these treatment regimens. So the first aspect that we can sort of have a look to when we think about behavioral management is the environment and supporting sociality of our animals. So when we look at increasing environmental complexity around our animals and supporting sociality, so supporting successful pairings over time and control and choice, we're looking at having, again, environments that foster species-typical behavior, that foster interest, and again, pair behaviors or group behaviors. One of the things that we know is that environments with very little stimuli can provoke sensitization to even the smallest variations, and that's been demonstrated, whether it's noise or scent. And this really has the potential to affect animals in a very exaggerated way. And we know it can underestimate treatment effect. And I think there's also been similar observations done nicely by Kelly Metcalf-Pate when we talk about SIV modeling, where we start to see difference in animals who have different social conditions imposed as well. So one of the things that I think is also relevant beyond just thinking about how does this environment and their control over it and how they experience it influence, again, the scientific outcome, which I just talked about, but it also allows us to have an evaluation of welfare that's very different from our typical ones, where we look at sort of traditionally negative welfare indicators, right? So we look at things that, again, may be atypical for the animals. Instead, we're looking at, in this situation like this, play. So it gives us a welfare assessment that's looking for a positive balance in our animals, which is also an exciting and different way to look at the animal experience. So one of the things that we aim to do here was really look at the impact of our most common. For most of us that are again, looking at safety and efficacy, that's typically sampling and dosing of our animals. And that can be either restraint-based, which we see most commonly used, whether that's sedation or physical restraint, which has the potential for stress or chemical confounding of our outcome measures, and is quite a bit less representative of what we'd anticipate in the clinical situation, where patients are not experiencing restraint for these types of common procedures, as compared with cooperative, right? So that's less commonly used from what we see when you look over the literature. It's still quite infrequent to see that animals are working cooperatively. And this requires training for cooperation, but also has less potential for confounding of outcome measures, and certainly is more representative of the clinical patient situation. And the reason that we were interested in this, of course, is because of the physiological effects of restraint-based sampling. So one of the major aspects of this, of course, is stress induction that's capable of affecting a lot of these downstream aspects of physiology, including immune status, and particularly for the focus of most non-human primate studies, being immunology-based because of the reasons that we talked about for model selection of primates. This raises a high level of concern. Chemical restraint, of course, also has the potential to introduce direct drug-related physiological effects. So here, what I wanted to talk about a little bit is again, the value of positive reinforcement training. So what you're seeing here is one of our cynomolgus macaques presentation to one of our caregivers for blood collection or drug administration. This is a practice session. And again, what we're looking for here is that roll-in period to do skills training and qualification. And they're going to learn things like injections, oral dosing, simple physical exams and vitals. This is a way to collect urine on cue. Of course, blood collection, dosing as you see, oral dosing in a reliable way, blood collection via heel stick or vascular access port, which we'll show in a moment. And for those of us doing metabolic work, extended conscious metabolic testing or fluid drug administration and hydration assessments. This also allows us a way to manage critical care for our animals. And why is this relevant? Because we certainly see that it mitigates the stress confounding and makes what we considered near clinical enrollment possible. And what you can see down in the corner there is you have this, this happens to be an intravenous glucose tolerance test. What we see is the comparison between ketamine, and that's in green, versus animals who are cooperating with a glucose disappearance rate calculated between 10 and 30 minutes. And what we see is not so much that the use of ketamine makes the curve abnormal, but it certainly depresses the glucose disappearance rate. So that, again, you introduce inaccuracy more than anything. Then you could look at the curve with animals receiving ketamine and look at that to be quite normal, but you're actually underestimating the glucose tolerance of that animal. One of the things that I like to point out, of course, is that our training is very feasible for an investment for most groups. So we average about 3 to 5 hours in total training time for our animals, with rhesus training out a little bit sooner than our cynomolgus macaques. And I also just popped in here the CIT-07, which is the clinical islet transplant study that was done. Just pulled out the inclusion criteria just to again remind ourselves what we would expect in the patient situation, that patients are able to comply with the procedures of the study protocol, that they're involved with intensive diabetes management. And so one of the things I think, again, we remind ourselves is that this improved adherence to the study protocol ultimately leads to better data. And certainly from the perspective of reduction of overall animal use, we see a reduction of dropouts, improvement of statistical power, and that, of course, increases our confidence that we've answered these questions more conclusively. So as we looked at our model influence on research outcomes and did a retrospective study looking at our animals that were under, for example, ketamine sedation versus cooperatively trained, what we could see very clearly is that even in a single event, that chemical restraint blunts serum cytokine expression. And again, when you're looking at the immunology aspects of these animals and trying to understand outcomes from the immune perspective, it becomes very concerning when you see, for example, in this case, blunting of multiple cytokines on a shared physiological pathway. So you see what is essentially a general systemic effect on cytokine expression between cooperative and sedated animals. So this was something that we found really interesting and very objective, again, in how chemical-based restraint was influencing our animals. And this, again, if you're interested in looking in more detail, is in scientific reports. The link is there. In the same way, understanding that, again, this is not just a sort of mandate to say, well, we should include both males and females because it might have some influence on outcomes. We see very clearly between males and females that there's a sexual dimorphism in cytokine expression patterns that really need to be addressed as you're thinking about your study design. So I wanted to share that as well as we think about how our design of experiment influences our outcomes. So one of the things that we know, and what you're seeing here is a rhesus male working under positive reinforcement paradigm for vascular access port access, is that we see some direct and indirect benefits that come from this, of course. So direct on study is that we have the ability to obtain physiologic data about the animal's health that we could not otherwise obtain, right? So these frequent serial checks that happen that are requiring some invasive or aversive handling couldn't be done in the same way if you needed to use chemical or physical restraint and require removal from their peer group, those sorts of things. So this really allows us to think very differently about our experimental design. So it increases the supportive care that we can do for our animals, and it really enhances well-being by bringing them closer to the clinical situation. So we can do things like rehydration, TPN, and again, as I said, monitor with a frequency that really allows us to intervene early and in a very effective way from the welfare perspective. Indirectly, what we see is the support in coping with the research environment. So you can't evidently predict everything that's going to happen on study, certainly, and what this allows us to do is have a general positive cognitive bias that we see from animals towards caregivers. So they associate these interactions with caregivers as rewarding. And what we can assume from that is it reduces the perception of pain, of course, but also allows us to, if we don't have something that we anticipated, be able to do that with an animal who's ready to cooperate. One of the next things we decided to really look at, having seen the cytokine data, and this is work, again, by one of my students, Scott Hunter-Oppler, that's having a look at the impact of cooperation versus chemical restraint during intensive sampling. So what we would see in the situation, again, of a typical tox study, for example. And what you see here is the timeline. So pre-sampling event for baseline health physiologic assessment, day 0 sampling and test article administration, and then relatively frequent sampling, followed by less frequent regular sampling for the duration of the study. And we had 6 animals in the group that was chemically restrained, 3 males, 3 females, and 6 animals in the cooperative group, so a total of 12 young adult monkeys. And this is a 42-day study. And what you can see, aside from what I've already shown retrospectively with the changes in immunology, is as we look at sort of, again, standard outcome measures that we would report in a regulatory situation, is we see incidence of nausea and abstinence. So again, things like low biscuit consumption and vomiting were significantly greater in animals sampled under chemical restraint compared to those who were sampled cooperatively. And this, of course, should come as no surprise to anyone, right, when you're using chemical restraint. But I think it's quite important that we show that this is significantly different between the two groups, and that, again, it's something that you need to be mindful of as you think about how it could be improperly attributed to the test article. So it is possible to increase our data accuracy and reduce the variability that we see by refining our model design. We've also previously published our work when we moved to, again, from the use of intensive chemical restraint and in combination oftentimes with these immunosuppression regimens and transplant, how you see actual pathological changes, for example, in the thymus, which again, you might expect, again, when you're looking at immunosuppression regimens, but you see that when you're working with animals who have been cooperatively trained, and that's the only difference, again, not the test article change, range, you can see a completely normal thymus. And again, it's incredibly important to really understand the differences that result simply from handling alone. And again, I like to show this, and we've shown this before in drug discovery today, the comparison between having a very minimal behavioral management program where you see that glycemic control in the top panel in A versus the second row where you have extended behavioral management, and you see the difference even in routine glycemic control, which is the primary outcome measure, of course, for us. And as you look to panel B, this is a reflection of weight between animals with behavioral management and those that don't have a full behavioral management program. And it's quite striking how you see the difference in weight loss, or essentially having animals really thrive under this regimen. And again, the difference being behavioral management. And we really aim to show that this is not related to our metabolic intervention, because as you see in panel C, the insulin exposure of the animals is very similar. So it's not a deficit in insulin. It's not a deficit in glucose disposal. We see, again, that there's relative little difference between glucose disposal. It's improved, of course, in the behavioral management group for the reasons that we know, as I showed earlier. But again, the real difference being the number of sedation events or number of restraint events that these animals were exposed to, making a big difference in primary outcomes on the study. So experimental designs can certainly be influenced by many factors. And we know that researcher safety in infectious disease studies or thinking about the ease in which we sample are big factors in how we set this up. But certainly some of the stress on research subjects related to these decisions are important to think about, because we know that it can ultimately confound study endpoints related to immune response and certainly safety, two of which we talked about as very objective examples today that are very critical in our ability to evaluate new drugs or devices. Behavioral management can foster resilience in our animals and certainly improves welfare. And I think it's important to always acknowledge that translational failures can happen despite our best, most rigorous science. And in using reverse translation or thinking about how we look at our clinical situation and what works, what doesn't work, and thinking through those refinements are really key to enhancing our modeling. And of course, deeply characterizing our models avoids our misinterpretation of study endpoints. So no matter what our study model design is, really understanding its influence on these outcomes supports our intentionality in how we select our models and how we design our experiments. And with that, I want to acknowledge the great team that was doing this work. In particular, I really want to call out work that was done by our students and residents who contributed to much of the new data that you see here, and of course the funding agencies that supported this work, in particular NIH and JDRF. Thank you.